All right, everybody. So today we're doing some genetics. Let's get into it. So nucleotides, we've talked about before. Nucleic acids are a biomolecule. DNA and RNA are what we're basically talking about. They're just repeating polymers of nitrogenous bases, which sounds super smart, but basically they're just these molecules that keep repeating over and over again. That's what a polymer is. Nucleoside, which we don't actually encounter too often, is just the base. And so you can see the top right corner here, that's your base. And then you're going to have some kind of a sugar. So usually it's a ribose. That's a ribose right there. And when we add on a phosphate, we call that a nucleotide. Now, we've encountered these before. Adenosine triphosphate is a nucleotide because it's adenosine, which is the base, with a sugar. And then there are three phosphates. In this case, we've got two phosphates attached. So just remember those bonds between those phosphates have a lot of energy. But in the case of DNA or RNA, those bonds are, we're going to actually kind of connect those phosphates to other bases. And then we're going to form this long polymer chain of nucleotides. So let's talk about nucleotides then. Those nucleotides we're going to connect, and one of the things I want to talk about a little bit is the actual numbering system that we use to talk about what direction your nucleotides are going in. And so we know, you may have heard, like cytosine and guanine and thymine and adenine, those are the C's, the G's, the T's, and the A's that comprise DNA and RNA. Now in RNA, we actually replace thymine with uracil. This is actually a uracil right here. I just forgot to replace thymine right here, but because I covered up the methyl group that's right here. But in DNA, we have C's and G's and T's and A's. And in RNA, we have C's and G's and A's, and we replace our T's with U's, but they actually look really, really similar. Now, how we actually number them is really interesting. So if you look at the ribose here, we've got 5 carbons. So 1, 2, 3, 4, 5 carbons, okay? And we attach our phosphate onto the 5th carbon. And so there's our nucleotide. When we connect nucleotides to each other, we take that phosphate and we attach it to the 3' carbon as opposed to the 5', which has the original nucleotide's phosphate on it. And then you just keep on stacking them. And it turns out the enzymes that build DNA or RNA, those are called polymerases. So there's a DNA polymerase that builds new strands of DNA, and there's an RNA polymerase that builds new strands of RNA. And it turns out they always take new nucleotides and they want to extend this chain, and they always attach them to this 3' carbon right here. Okay, so they're always tacking on this phosphate onto this 3' carbon, forming this bond, and then we just kind of keep on stacking them in that direction. So we're going from the 5' direction towards the 3' direction. So we always build 5, 2, 3. Now in the case of DNA, we have 2 strands. And we're going to talk about that structure, typically that double helix, but RNA would just have one of these strands, but they still have that directionality of going from a 5' to a 3'. And then on the other side, they would go from a 5' to a 3', but in the opposite direction. So if you look here, we've got our 5' carbon going towards our 3'. We would always build in this direction on this strand. Okay, so we'd add on another nucleotide here, another one below that, another one below that. On this side, it's the opposite because everything's kind of flipped and they go in the other direction. And so we've got our 5' carbon here, 5, 4, 3, 2, 1. And so there's our 3' carbon here. So we go from 5 to 3 in this direction. And so we're still going from 5' to 3'. It's just in the opposite direction. Now we need to talk a little bit about the differences between DNA and RNA. Now I already mentioned RNA has uracil instead of thymine. The only difference between those things is that thymine has an extra methyl group, which is just a carbon with 3 hydrogens attached, sticking right here. You can kind of see I have a little edge right there where I covered it up with like a little white square right here. So this is uracil actually. So if you stick a little methyl group on there, it becomes thymine. That's the only difference between thymine and uracil, except all of the nucleotides in RNA have an extra hydroxyl group. So they have an extra oxygen on their number 2 carbon right here. So you can see I stuck this on here. So this would actually be a uracil, and it's an RNA because it has a nucleic acid. It's ribonucleic acid here. If I remove this oxygen, if I deoxy it, it becomes deoxyribonucleic acid, the ribo referring to this sugar, which is called ribose. So if I take this oxygen off, it becomes deoxyribonucleic ribonucleic acid. This is a nucleic acid. And so that's where these names are actually coming from. And so here are just our structures of the riboses of either DNA. Okay, we've got 1, 2, 3, 4, 5 carbons, just like before. And if I take that oxygen off here, so there's the oxygen in RNA, so it's just a normal ribose with that oxygen. If I deoxy it, now it becomes deoxyribonucleic acid. And now you know when you hear DNA thrown about or RNA thrown about, because like the COVID vaccine is an RNA vaccine, well, now you know that's the difference. It's really not that different. It's just different enough that we can tell the difference if we really look at it. But for the most part, how we actually make our nucleotides and how we actually connect them is the same. And so connecting nucleotides is really interesting. And they form phosphodiester bonds between the nucleotides. Now, this is just a different picture, but you can see here we've got our nucleotide. Okay, there's our base with our ribose. Okay, 1, 2, 3, 4, 5 prime carbon. There's our 3 prime carbon. And you can see we're going to build a bond between this phosphate group on the next nucleotide and our little ribose sugar right there, which is kind of fun. And it's a condensation reaction. If you have not taken organic chemistry, that is perfectly fine. It's really, really simple. We're just going to take this hydrogen and this oxygen, and we're going to remove them and then have this phosphate kind of bond to this oxygen right here. So this bond right here that's stuck to this oxygen, we're going to replace it with this oxygen. And so now we have a bond between those. And so you can see we've got our phosphate here with this oxygen right here, and it's bound to this 3 prime carbon, okay? And so we always build 5 to 3. So we would tack on our next nucleotide to this 3 prime carbon down here. It's called a condensation reaction because we're actually generating water. There's a hydrogen, 1, 2 hydrogens, that's H2, and O, so H2O. So we end up with water. The enzymes that do this are called polymerases, as I mentioned before. There are DNA polymerases that will put new nucleotides that are deoxyribonucleic acids. So these are oxynucleic acids, so these would be RNA. But if I removed this hydroxyl group here and just put a hydrogen, it would be deoxyribonucleic acid. Let's get talking a little bit about the secondary structure that we see in DNA, because we want to start looking at those double helices, right? So we know that there's A's and T's and C's and G's, and so it turns out they have different structures. So adenine and guanine are bigger, and you can see them here. So they have 2 rings. Okay, so there's 1, 2 rings for guanine. There's 1, 2 rings for adenine. Those are called purines. And then the pyrimidines are the others. And so we've got single rings for those. So there's cytosine, there's thymine, and then if you remove this methyl group right here, it becomes uracil. Okay, that's really the only difference besides that extra hydroxyl group from over here. And what's really, really important is that G's and C's will bond together and form 3 hydrogen bonds between them. And A's and T's will bond together and they will form 2 hydrogen bonds between them. So it's really important to know that guanine will only ever match up with cytosine. At least it's only supposed to match up with those. There can be mistakes that happen. We'll kind of talk a little bit about that when we talk about mutations and mutations repair next week. So G's match up with C's and have 3 bonds. T's match up with A's and will have 2 bonds. Why is that important? Well, the more G's and C's you have, the more bonds that you're going to have, right? So there's always 2 hydrogen bonds between every nucleotide, but G's and C's have an extra one. They have 3 hydrogen bonds. So the more G's and C's you have, the more tightly held your 2 DNA strands are, because this is one DNA strand, and this is the other DNA strand. You can see we're still progressing. We have our 5' to our 3' end. Notice it's an arrow, because we would always build this direction with this strand, and we would build in this direction with this strand. And so we know that G's and C's always bind together. They have 3 hydrogen bonds. So the more G's and C's you have in your DNA, the stronger or the more tightly bound those 2 DNA strands are held together. That's actually one way that we can separate certain species of bacteria. We know that entire genuses or entire phyla of bacteria are called high GC content bacteria, in that their DNA has a lot of guanines and cytosines. Therefore, their DNA would technically be harder to pull apart. Now let's get into these double helixes. So We would call this our tertiary structure of DNA. So the primary structure is just the order of nucleotides. And then the secondary structure would be having two strands stuck together. And then we start wrapping them around each other. DNA is a double helix, okay? And so that means we end up with two grooves, our major groove, which is a little bit bigger. Okay, so you can see it here. It's 22 angstroms wide. And then we have our minor groove, which is only 12 angstroms wide. Why is that important? Well, we have lots of enzymes, which we're going to talk about in a little bit, that need access to the DNA, to that DNA code. And so we have our major and minor grooves, so that allows our major groove to be a little bit bigger and thus allow access to those nucleotides by proteins or enzymes that need access to them. And so if I want to go in and create a messenger RNA off of this DNA strand, which is a process called transcription, RNA polymerase is the enzyme that does that, and it needs access to those nucleotides. And so a lot of these enzymes will actually go through the major groove, and they kind of slide along the minor groove. Now, of course, there are exceptions, but what you gonna do? So if that's our double helix, we can also take our DNA strand and we need to keep it nice and organized. So one of the most important things that biological systems need to do is keep their DNA nice and organized. If you don't keep your DNA organized, it can get all wrapped up. DNA is really sticky in and of itself, and so it can get wrapped up in stuff, it can get damaged, and we don't want that. And so you can see here, there's a few different kinds of histones, but basically what you're doing is you're taking your DNA strand, which is just like a long rope, and you're wrapping it around this nice solid protein thing that you can then wrap around other solid pieces of protein, and we're able to establish a higher order level of structure. It allows us to make sure that our DNA is much more organized and easy to access, and so we can close off certain portions, and we can make it really, really tight, okay? Or we can open it up and free up that DNA, which makes it easier to transcribe or replicate. That's called epigenetics, which we'll talk about later. And so what you're seeing here is we've got our DNA wrapped around our histones, and it can be nice and loose like this. This would be really easy to get an RNA polymerase in here to actually go in and transcribe a gene. But if it was really dense like this, it would be really hard to get the enzymes that we need inside here to actually bind to the DNA strand. And that would be really hard to make a messenger RNA from this segment, which we could then turn into a protein. Something I've actually seen in textbooks, a question that was asked on an exam that I had seen before was that bacteria do not have histones. Technically, that is true. They have histone-like proteins. So it's one of those technicalities. Bacteria also have big chromosomes. They're not as big as ours, absolutely. They can have massive numbers of nucleotides. And so bacteria do need to keep their DNA and their chromosomes, which is just when you start combining all of these proteins and DNA together to form these giant chromosomes, which this should look like something you've seen before, right? So our bacteria also do that exact same thing. Now, it depends on what stage of growth they're in, which we've talked about before. If they're in log phase growth or exponential growth, then they're going to be doing a lot of replication. They're going to be doing a lot of transcription to actually make proteins. And so you can see on the left side here, there's a lot going on. There's a lot of proteins that are around. There's a lot of histone-like proteins that are around, giving it some structure. And on the right side here, you can see during stationary growth, so we're right here, during stationary growth, the scaffolding isn't necessarily as technical or as extensive because there's not as much going on, so you don't need as many histone-like proteins. Now, if we look at either prokaryotes, which are bacteria or archaea, and eukaryotes, our chromosomes definitely are more complicated in eukaryotes, but that makes sense because our cells are more complicated. They're a lot bigger, they have a lot more things that they need to do, and so prokaryotes are quote-unquote simpler. Now, that doesn't mean that they're not immensely complicated and more complicated than any human has ever understood. There is one thing that I don't like about this diagram. I like that it's depicting that there's a lot more DNA inside of a eukaryotic cell's nucleus. This would be like the nucleoid of a bacteria. But they're showing the histones here. There are histone-like proteins associated here. And so I just kind of wanted to show this to show that even on these nice diagrams, they're missing something. Some important terms to make sure that we're clear on is our genome. So the genome is just all the genetic information inside of a cell. That includes all the genes, all the coding regions, all the non-coding regions. Genomics is the actual study of genomes and genes and things like that. Chromosomes are just a higher order level of structure. So if you've got your DNA, you sort of organize that into chromatin, which is your DNA plus your histones, those proteins. And then those really large complexes we call chromosomes. Most bacteria have 1 or 2 chromosomes. Archaea can have 8 to 10-ish, but humans and other eukaryotes can have tons of chromosomes. Now that's going to lead us into talking about how we actually make new copies of our chromosomes, because cells are always trying to replicate. Every biological system on the planet is trying to make more of itself. That's just what they do. And so we need to talk about, how do we make more DNA? How does a DNA polymerase actually build new strands of DNA? And so in order to show you how that happens, it's a very physical process. It's really nice to have a good animation. And so I want to cut to this animation that's going to really depict all the enzymes, or most of them, and it's going to really show us what's going on.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So, the way I like to demonstrate DNA replication and what helicase actually does to the double strand is I like to use a rubber band. Okay, so I've got a rubber band here, and I'm going to twist it around so you can see I'm getting some twists, and this is going to look like our DNA helix. So it's kind of just the twists that we see in our DNA helix. So you can see it's kind of twisted around there. So we've got our double helix. You can see it's just all bunched up. And so what's going to happen is we're going to use our helicase, and we're going to use our helicase to try and open up the strand. I don't usually use this thick of rubber band. Okay, so we're going to open up a little hole there. Okay, so I'll put my finger in there. So this is where we actually have our helicase initiating replication. Okay? And so it's going to open it up in we're going to have two helicases going in each direction, but we're just going to follow one direction for now. And so our helicase is going to open it up, and it's going to start pushing this way. And I want you to just watch what's happening, okay? So it's going to push, it's opening up, which is great. So it actually really looks like this. And then as I get closer and closer to this end, you can see what's happening. All the knots that were, all the twists and turns that were in here are now being slid to this end. And you get what we call supercoiling, as you can see here, is fixed by topoisomerase, which should sound familiar. You may have heard of it, called DNA gyrase. So when we've talked about quinolones and fluoroquinolones during antibiotics, fluoroquinolones will disable topoisomerases. Now, there are a couple different kinds of topoisomerases, so we don't have to worry about quinolones actually hurting ours, because bacterial topoisomerases are kind of different. They actually serve a sort of a different function, but basically what they do is they cut the DNA and let it kind of untwist, let that supercoiling untwist. And what does that do? Well, that kind of just relieves that tension and allows the helicase to keep opening that replication fork. And so let's talk about that replication fork a little bit. So in a prokaryote, so like in this example, we've got an E. coli, they're going to have one, maybe two origins of replication is what we call it. And that's just that spot where the DNA strands have opened up and you have two replication forks going in either direction. And you can see this in animation here where we're kind of separating out these two strands, these two circular chromosomes. Eukaryotes also have origins of replication, but eukaryotic DNA and eukaryotic chromosomes are huge, and so you need to have hundreds, thousands, hundreds of thousands of origins of replication just to finish replicating in time to actually undergo mitosis. So I want to make sure it's very clear, so I'm going to show a picture of an origin of replication. Just like we saw in that video, they did a really good job of explaining the leading and the lagging strands. And I just want you to be familiar with the directionality of DNA once again. So remember, 5' to 3' is the most important. So we always build from the 5' towards the 3' direction. So remember, that carbon, that 3' carbon that's on your nucleotide, that's where we attach the phosphate from the next nucleotide, which is attached to its 5' carbon. And so we can just keep on stacking them in the 5' to 3' direction. So you're always going towards the 3' direction. So when we open up our replication fork, we always have a leading strand going this direction, building 5' to 3. And we always have a lagging strand that's trying to make up for that. But on the other side of that origin of replication, we have another replication fork that's the opposite. And so, yes, the leading strand of one side also has a lagging strand kind of catching up to it. And so usually we're just talking about one replication fork in an origin of replication, just to keep things from getting too confusing. But there are two replication forks. Important term that gets tossed around and is talked about in that video are Okazaki fragments. And these little guys here are those Okazaki fragments. And they're just really small pieces of DNA. So if you had a cell undergoing replication and you pull out all of the DNA strands, you would find these little pieces. And a researcher named Okazaki found them for the first time, and so we call them Okazaki fragments. Now I want to talk about what this actually looks like. And so a lot of times the diagrams that we actually use are rather simplified. And so I want to show you what it actually looks like. And so the quality of this picture isn't the highest, but it's probably one of the best ones that I've seen that keeps it relatively simple while still maintaining some of the complexities. And so what I want you to see here, there's your topoisomerase, or in this case they're saying DNA gyrase. Okay, we just talked about that. Here's your helicase, which is going to sort of unzip the DNA. And then you can see we have our primase here, right? That has to lay down that RNA primer. And then that is where our DNA polymerase, or in this case DNA polymerase 3, is going to attach sort of, it's looking for that RNA primer, it primes it, and it starts replicating and adding a new DNA strand. And so up here, we're just looking at one replication fork. So this would be our leading strand, right? So we're building 5' to 3, just like in the other diagram. And we're building 5' to 3 this direction, and so it would keep opening up this way and just keep on chugging along. You can see that this whole complex is sort of like held together, and so they're not these separate enzymes that are just kind of coming together just randomly. They're all sort of contained and held together by a bunch of different things. We call it the clamp loader, which isn't that important. It's just the scaffolding on which all of these enzymes exist. And so we're building from 5' to 3' on this guy. And so there would be like a primer somewhere over this direction, but it's going that way. And then you have your lagging strand on this side. And so you can see we're still using the same DNA polymerase. It's still starting at the primer and moving towards this direction. And it's going to encounter this next primer. And something that you don't usually get in a lot of micro classes is that the exonuclease would come in and eliminate this. They did talk about that. And then you have another DNA polymerase come in and replace those nucleotides where the RNA was, because we know RNA and DNA is different, right? So that RNA gets replaced by a different DNA polymerase. It's called DNA polymerase 1, at least if we're in bacteria. And so that's just kind of a subtlety, but it kind of explains, like, it's not this exact same enzyme coming back and fixing that problem. It's a different enzyme. There are lots of different polymerases. There are lots of different enzymes associated with this. This actually covers most of them, but there are some complexities to this. A lot of these enzymes have multiple domains, which means they have different activities within their own protein structure. But for the most part, this is exactly what's going on. Now, this is looking at an E. coli. That's a very well-studied organism. Eukaryotes have different enzymes. We call them different names. Sometimes different cells do different things. For instance, in the mitochondria, we have our own mitochondrial chromosomes as opposed to the chromosomes that are inside the nucleus. And so we have our own DNA polymerases inside the mitochondria. So that's really interesting. And so there are lots of other examples of basically eukaryotes are more complex and we have more enzymes, but they're still doing the same basic stuff. A DNA polymerase is going to make more DNA strands. Something that video talked about was semi-conservative replication. And what does that mean? Well, we need to talk about that. So before we could actually look at DNA inside of a cell, there was a debate. Nobody really knew exactly how DNA was replicated. And so they did an experiment. And so they took the original piece of DNA and they coded it in radioactivity so we could track it. And then they allowed the cells to undergo replication. And basically what happened was they would find half the radioactive DNA in one of the daughter cells and half in the other. And then they would allow replication to happen again. And then, so now they have 1, 2, 3, 4 cells, and they still only have 50% of those daughter cells have that radioactivity in their DNA. Now, what does that tell us? Well, that tells us that each of these parental strands, because there's 2 strands of DNA, each one is being pulled apart and then having a new strand added to it, a complementary strand. So you pull apart one of these parental strands, pull it apart, and build this new strand alongside it that matches up, and that's not radioactive, right? Now, up until that point, there was a debate about, well, how do we really know if that's the case? I mean, that could be the case. But another case could be, what if you take your DNA strands and you pull them apart and you make new complementary strands, and then you put those 2 original parent strands back together and those 2 new daughter strands, and those end up in each separate daughter cell. But we know now that's kind of silly. That's not how it happens. We know that DNA is built, so the green one would be a parent strand, and then the new daughter strand would be attached to it. And it seems kind of silly now to think about that. Why would you pull them apart? They're like stuck together now. And so, well, it took a little while in some experiments, but we figured out that that actually is how it happens. The dispersive model was kind of crazy. It just said that it was sort of random, that you would like take pieces of each one and then stick those together with other pieces, and that was kind of nuts. But semi-conservative replication is what actually happens. And the conservative is just referring to the idea that it's conserved. That's what conservative means, right? So are they conserving those strands? Well, half the parental strand is conserved and the other half goes with the other daughter cell. And so half of it is conserved and the other half is made complementary and it's new. So DNA replication we need to talk about, and we're going to kind of zoom out a little bit and talk about the cell cycle. Now, if you take a genetics course, we talk about the cell cycle all the time. We spend a lot of time on it, but the cell cycle is important. We need to talk about it just a little bit. And so in the cell cycle, basically we're gathering up all the nutrients and all the things that we need and doubling our organelles and making sure that we have everything to make 2 fully functional daughter cells. And then we're going to actually do the process. And so the cell cycle are the things that we need to do to get ready for that. The cell cycle functions in a couple different phases. There's the G1 phase, or the growth 1 phase, and that's the main growth phase. It's the biggest one. You're going to make more organelles. You're going to make your size huge. You're going to be doing tons of metabolism. You're going to be making proteins up the wazoo. And then you enter the S phase, and the S stands for synthesis. That's where replication happens. So when we're talking about this process, that's occurring inside the synthesis phase. Now the nucleus is still there, you still have your chromosomes inside the nucleus, but you're doubling your DNA because you want each daughter cell to have a complete copy of your DNA. That's really important. Because if you remember back to cell theory, cells can only come from other cells, and they have to keep their DNA, and that's where this comes from. Then you enter the G2 phase, which is definitely not this big. It's way smaller than this. G2 is just a little bit of additional growth. It's basically the pause before you enter mitosis, which mitosis you probably have heard of, and that's just the process that takes us from one cell and splits it into two daughter cells. Each phase of mitosis has a specific purpose. It's really a continuous flowing phase. We separate them because we're humans and we like things nice and organized, but really it's one continuous process. So we go from prophase, which is where our chromosomes condense down. They don't normally look condensed like this, okay? But in the prophase, they do condense down, and the nuclear envelope dissolves because we need to pull apart sister chromatids. So sister chromatids occur during S phase. You have a chromosome from mom and a chromosome from dad, and your daughter cells, when you're going to split a cell in half and make two new daughter cells, they each need to have a copy from mom and copy from dad. So you need to have 2 copies from mom and 2 copies from dad, because one of those has to go into each of those daughter cells. And so we need to pull those apart. Those are called sister chromatids. And you can see them right here. These would be sister chromatids. So there's one dark one here and one dark one here, and then there's a light blue one here and a light blue one here. This is from mom, this is from dad, or vice versa, it doesn't really matter. And so what we're going to do is during metaphase, because during prophase we condense them down, during metaphase we line them up along the metaphase plate, and then we're going to pull them apart during anaphase. Okay, so there's one from mom, one from mom, one from dad, one from dad, and you can see we're going to end up with, during telophase, we end up with one from mom and one from dad on one side, and one from mom and one from dad on the other side. Sometimes the process of cytokinesis is listed as occurring after telophase. Technically, it can start occurring at the beginning of telophase and then kind of proceed through telophase. Cytokinesis is just the full separation of the daughter cells. Prophase, metaphase, anaphase, telophase, those are really more referring to the chromosomes. We're sort of using those as our way to track what's going on inside the cell. Cytokinesis is really looking more at the cell level and saying, okay, even at the end of anaphase and the beginning of telophase and all the way through telophase, those two cells are really starting to become their own individuals, and that's cytokinesis. So now that we've talked about how we make more DNA, which is replication, we need to talk about the central dogma of molecular biology, which is where we go from DNA to RNA to protein. So the central dogma is really straightforward. It's the process of transcription and translation and the in-between steps. So DNA is deoxyribonucleic acid, that's what we've been talking about, and we're going to make our messenger RNA using our DNA as a template. Okay, that process is called transcription. If you transcribe something, you're usually staying in the same medium. So for instance, if I had a master blueprint, let's say I'm an architect, I'm not going to take those master blueprints out to the place where they're building the building that I designed. I'm going to make a photocopy, right? But it's still going to be a picture of those blueprints. And so that's what transcription is. It's kind of a photocopy of your DNA, because your DNA is very, very valuable. So we make a little copy of it called messenger RNA. It's still the same thing. It's still a piece of paper, if you will. It's still nucleic acids. And so what we're going to do is now we're going to take those nucleic acids and give them to our builder, who's going to translate it into a new language called amino acids. That's what proteins are made of. So we're going to go from DNA to RNA to proteins. So let's zoom in on each of these things. So first, we're going to talk about transcription. So transcription is going from DNA to RNA. And basically, the initial start of it is very, very similar to replication. We have to open it up. And we have 2 strands of DNA, so remember that. So what we're going to do is we're going to have our coding strand, okay? So we've got our A, T, G, A, T, C, okay? It just keeps on going. And we're going to open it up. And so the opposite strand of DNA would be complementary to that, right? So we'd have an A here, so therefore you'd have a T on the other side. We have a T here, so you'd have an A on the other side. You have a G here, so you'd have a C on the other side. Now, when we make our messenger RNA, we want our messenger RNA to look exactly like our coding strand. And so here, if our coding strand is ATGA, then we want our RNA to look like ATGA. Now, one of the tricks is that T's don't exist in RNA, they're U's. But remember, they're really similar, they're just lacking a methyl group. And so it looks like almost exactly the same. So we've got AA, UT, right? T's and U's are interchangeable. GG, AA, UT, CC, UT. And so we're maintaining that exact same order. So your messenger RNA will look exactly like this coding strand. The other strand is called the template strand. And so that's the template strand because we're using it as a template to actually make the messenger RNA. And so if my coding strand is ATG, then its complementary template strand is TAC. And I can use that to build my RNA. So if I have a T, then the only nucleotide that would match up with a T is in A, right? And if I have an A, the only RNA nucleotide that would match up with an A would be a uracil, okay, because that looks like a T. And if I have a C, it has to be a G. And so this template strand, my RNA doesn't look like the template strand, it's the opposite, but it does look like the coding strand. I have another diagram here that sort of shows what's going on, where we kind of zoom in. There's a few terms that get tossed around. I like coding and non-coding. I was actually taught sense and antisense. I don't love those terms because they don't really explain what I just explained super well. But the coding strand is the sense strand. It makes sense. That's not what it's referring to, but that's how I like to think about it. So it's sense and then anti-sense. So those are the opposite. So the non-coding strand or the template strand, this strand, is the one that I make my messenger RNA off of. The enzyme that does that is called RNA polymerase. Yes, it's a polymerase. It just makes RNA. We name our enzymes off of what they do. DNA and RNA are polymers. So this is a polymerase. And this one happens to make RNA. Now, where does it know where to start? That's a great question. There's a variety of ways that we do that, but basically you have what we call a codon. So let's talk about codons. Codons are a series of 3 nucleotides, so we're starting to break our DNA and our RNA into smaller pieces, okay, smaller usable pieces. And what we've discovered over the last century, basically, is that we organize our DNA and our RNA into 3 nucleotide units. We call those codons. And each codon codes for 1 amino acid. In humans, there's 20 amino acids, to be slightly generic about it. There's a couple weird random ones. But basically, I take every 3 nucleotides, and that equals 1 amino acid. And so then I move to the next 3 nucleotides, and I put a new amino acid. And then I move another 3 nucleotides, and I make another amino acid. Ribosomes are what are actually going to be doing this part, okay? That's what ribosomes do. But your RNA polymerase is what actually makes your messenger RNA. The order of nucleotides is very, very important. Now, there are lots of ways to teach this, but basically you just have your messenger RNA and you can use that to figure out what amino acids are going to be put in order. And there's a couple different styles of charts. I really like the circular style. And so I have 2 versions here. So this would be like from your DNA coding strand, okay, which I talked about. This would be from the messenger RNA, which would be looking exactly like the coding strand, except instead of thymine, it would have uracil. But you're either given the DNA strand, A's, T's, C's, and G's in order, or you're given the RNA strand, A's, T's, C's, or I'm sorry, A's, U's, C's, or G's in order. And if you know every 3 nucleotides is a codon, well, then you can just say, okay, well, what are the first 3 nucleotides? Let's say it's UUC. And so, okay, I know that's RNA. So I would say, all right, I'm going to start in the center here. So I say U, uracil. U is the next nucleotide. And C, that codes for the amino acid called phenylalanine. Okay, that's pretty straightforward. Let's try it with a coding strand from DNA. Let's say CAG. All right, so we start in the center here. So I start in the center, there's C, A, G. Codes for glutamine. All right, that seems pretty straightforward, right? There are start codons, that's generally methionine, which is AUG. So you can see over here we've got A starting in the center, U, G, codes for methionine. And then you have to tell the RNA polymerase where to stop making the messenger RNA. And that is called a stop codon. There are 3 of them. There's one over here. There's UGA, UAA, and UAG. Those would all tell the RNA polymerase to stop. And then you've made your messenger RNA that can then leave the nucleus and head out to find a ribosome where we can then do this process. This process is called translation. Translation is where a ribosome grabs onto a messenger RNA and then makes a new polypeptide chain. And so you can see that's happening here in the bottom left corner. This ribosome is making this long chain of amino acids based on the codons of this messenger RNA. And in this case, this ribosome is now going to go and join up with the endoplasmic reticulum, this would be the rough ER, and it's going to sort of push this polypeptide chain into the endoplasmic reticulum. So this might be secreted or it might stay stuck in the membrane. But ribosomes are really, really interesting. We've spent some time talking about ribosomes. There's lots of antibiotics that affect ribosomes. But ribosomes engage in translation. And so to summarize this big process, here's just a nice picture where we're going from our DNA, we're transcribing it, making our messenger RNA. We're about to talk about how we process our messenger RNA, and then it's going to leave the nucleus, and then it's going to be translated. And then once we actually make our protein, that's where in the endoplasmic reticulum and in the Golgi body and in some vesicles, we can do different kinds of modifications to actually end up with the kind of protein that we want.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Now, I'm talking a lot about eukaryotes. Bacteria also do this, but remember, bacteria do not have a nuclear membrane. They don't have a nucleus. And so they can do something really, really cool. We call it polyribosomes. And polyribosomes are where we can actually have our DNA strand, so that's in blue, and we have our RNA polymerase that makes messenger RNA, right? And so you have your messenger RNA that's in red, coming off of here. And we know that messenger RNA goes to ribosomes and they translate, right? They make a amino acid chain. And so in this case, we see these things called polyribosomes in bacteria, where we have our DNA and our RNA coming right off of it, and there's no nuclear envelope to separate our ribosomes from your actual DNA. And so your ribosomes can get up close and personal with the DNA. And so as soon as that messenger RNA comes off of that DNA, it can start translating it like that. It's very, very interesting. So the last thing I want to talk about is how we actually control gene expression. So a gene is just a piece of DNA that codes for a messenger RNA that can then go and be made into a protein. That's just a really simple way of thinking about genes. Genes are not just their coding region, they're also the other regions that flank that region. And so you can have downstream flanking regions. So that would be beyond, if you're going from 5' to 3', right? So my RNA would be built from 5' to 3', okay, off of the template strand. And so when I actually do that, anything downstream is called downstream, and then anything upstream is called upstream. And there are lots of things, especially upstream, because this is the region where your RNA polymerase is going to kind of look around and look for the promoter region. There are specific pieces of promoters, of promoter DNA regions that tell RNA polymerases where to bind. This is also where transcription factors, which you may have heard of before, those are proteins that promote transcription. They're transcription factors. Some of them suppress transcription, but a lot of them promote it, and they bind to the promoter region. It's like it was named on purpose. Now, your genotype is the actual expression of genes. And if you take a genetics course, we end up talking about Mendelian genetics and non-Mendelian genetics, and that gets into genotypes and phenotypes. But genotype is just what genes you have, and then the phenotype is the actual physical expression of those genes. That doesn't have to be, like skin color, hair color, things like that. It could be just any kind of protein that gets expressed. So let's say we undergo transcription, and we make our messenger RNA. So here's our nice messenger RNA down here. And so what we need to do is a process called splicing. We need to modify our messenger RNA so that it fits the way that we want it to, or that it's going to code for the thing that we want it to. And so splicing is a really interesting process. You may have heard of this before, you may have not. So in splicing, we take our really long messenger RNA and we cut out sections that we don't want. Those are called introns. So we splice out the introns and we leave behind the exons. And so you can see here, we're going to keep this dark region here and this lighter region here and this darker region here, and we're going to stick those together and cut out the introns. You don't have to always do this the exact same way. You can cut out different introns, and we call that alternative splicing. This is how you can make multiple types of proteins that have similar domains, but have some difference in structure. So for instance, let's say this region right here is a nice region that likes to stay embedded inside membranes. It's got a really hydrophobic portion. And then the rest of this is the rest of some kind of surface receptor that's on the surface of a cell. Well, maybe I want to make my surface receptor look a little bit different. And so maybe I want it to grab onto insulin as opposed to testosterone. And so in this case, maybe if I keep this piece and this piece, this portion of the protein that this will eventually code for will grab onto insulin. But if I get rid of this one, make this an intron and make this an exon, maybe I can bind to testosterone. I completely made that up, but hopefully that concept of alternative splicing makes sense. We have lots of different ways of transcriptional control. Most of your genes are on all the time. That's called constitutive expression. It's all the stuff that just needs to be made all the time. But we don't always do that. Inside of us, we're very complicated creatures, eukaryotes. We use lots of transcription factors. We use lots of signaling molecules to tell ourselves what genes need to be turned on and which genes need to be turned off, because we have organs. We're just really complicated. Now, when you actually look at bacteria, they're a little bit simpler, but we need to talk about how they actually do some of their levels of control. They're called operons, and that's the first level of control I'm going to talk about. Then I'm going to hit epigenetics, transcription factors, and RNA interference. It's actually going to go pretty fast. But so we're going to talk about operons first. So operons are just groups of bacterial genes that can either be turned on or turned off. If they're usually on, i.e. constitutively expressed, but we could turn them off if we wanted to, we call that repressible. It's like if you have a really bad memory and you repress it in your brain. Okay, the memory is there, but I can turn it off. Now, that's as opposed to something inducible. So if I had a set of genes that are usually turned off, but some situation comes up where I need that thing, I'm going to induce those genes to make their proteins. And so we call those inducible operons. There's a big example of each of these. A repressible operon, the classic one, is called the trip operon. That's one that's always on but can be turned off. So it turns out that cells need tryptophan. So tryptophan is an amino acid. It's on the circle chart that I just showed a minute ago. If you don't have enough tryptophan in your cell, then you can't make your proteins because you need tryptophan to make proteins. And so we have enzymes that can make tryptophan. That's what the trip operon, the TRP operon, is for. And so it's normally on. Like, you need to be making tryptophan all the time. But it is a repressible operon. These genes can be turned off because, for instance, let's say that your bacteria goes and hangs out somewhere where there's lots of tryptophan in the media. Well, if there's lots of tryptophan around, why would I waste my time making the enzymes that make tryptophan? I have a bunch of it sitting around already. And so I need to turn that off. And we use what's called a repressor. A repressor is a protein that will bind to the operator, which is just a region up in front of or upstream of the actual operon, and it binds to it and keeps the RNA polymerase, that's this wonky looking thing here, it keeps the RNA polymerase literally from binding to this promoter region, which we talked about promoter regions just a second ago. And so the promoter region has this operon on it, and if this repressor grabs onto it, then we can't transcribe these proteins anymore. And we don't want to because we don't need to be making tryptophan. Now, once the tryptophan goes away, then it's no longer grabbing onto this repressor. It's not making it work. Okay? That's what's happening, is the tryptophan bound to the repressor and made it stick onto this actual chunk of DNA here. So it falls off when there's no more tryptophan around. And so you can start transcribing again. So that is a repressible operon because I can turn it off, but normally I want it on. That's opposed to inducible operons. And I kind of like inducible operons a little bit more because they kind of make sense. They turn on if you need them. And so, for example, if you have an E. coli, it likes to eat glucose. Glucose is a monosaccharide. It's really, really straightforward. We've talked about metabolism before, and we usually think about the breakdown of glucose. You know, you're going to undergo glycolysis and then send that stuff into the citric acid cycle. And so it's preferred over things like lactose. Lactose is a disaccharide. It's this is an oversimplification, but it's like two glucose stuck together. So I have to do more work to actually break it down. In order to do that, I have to use an enzyme called lactase. Yes, lactase, that same thing that we talk about, like if you're lactose intolerant, there you go. You need lactase in your gut to actually break down lactose. And so bacteria, kind of like us, prefer to eat glucose over lactose. So if glucose is present, then why would I make the enzyme that breaks down lactose? I don't need to. But if there is no glucose around, but there is lactose, then I'm going to make the enzyme that breaks down lactose so I can, keep living and making energy. And so in this case, normally my repressor is bound, okay? So it's stuck onto that operator. See, there's the operator in my promoter region, okay? And so I've got my repressor, it's bound here. And because there's no lactose around, then why would I make the enzymes that break down lactose? There's a bunch of enzymes that actually make lactase. And so why even make it? Who cares? But if lactose is present, it binds to the repressor and makes it fall off, which is the opposite of the trip operon. In the trip operon, when tryptophan is present, it binds to the repressor and makes it stick to that operator region. As opposed to this one, it's normally stuck to the operator region, but when lactose shows up, it falls off. And then my RNA polymerase can come in, it can bind to it, and then it can transcribe, and then we can translate those enzymes, and I can break down lactose. So those are bacterial operons, pretty straightforward. Those are the two classic ones that every microclass has to talk about. A couple different levels of control. I mentioned earlier epigenetics. So epigenetics is the density of your chromatin. So we talked about the structure when I wrap my DNA around histones. And if the histones are really tightly wrapped, it's really hard to transcribe them. If they're really loosely wrapped, then it's much easier to transcribe them. If I add methyl groups, okay, so that's just a carbon with some hydrogens. If I add methyl groups to my histones, they kind of tighten up. If I add acetyl groups, there's an acetyl group. If I add acetyl groups to my histones, they kind of open up. And so however much methylation or acetylation there is, I can control how tightly wound or loose my chromatin is. Those are called epigenetic controls, and that's one way that we can control how much of a specific protein actually gets made. So the last thing I want to talk about is this concept called RNA interference. This is another level of control. Basically, if I have my messenger RNA, I'm going to take it, it's really long, and I'm going to send it to a ribosome, and I'm going to translate it into a protein. Okay, that's fine. Well, what if you made too many of those messenger RNAs? Or maybe the protein that you're about to make from that messenger RNA is a protein you don't want anymore. So instead of going through that whole process of making a protein, you got to fold it, and then you have to get rid of it. That sounds like a pain. And so what we can do is we make these little RNAs inside the nucleus. They're called interfering RNAs. They're small RNAs. They're really, really small. And what we do is we chop them up, we get them ready, and what they do is they sort of act like a little guide. And so we take these RNAs, either microRNAs or small interfering RNAs, and we send them into this complex called RISC. And the RISC complex then goes around and it sort of uses it like a little key. And it looks for other RNA, okay, mainly messenger RNA, and it's looking and it goes and finds messenger RNAs that are just floating around in the cytoplasm, and it sticks its little guide RNA onto it. And if it's microRNA and one small region of the microRNA matches up with that really big sequence of messenger RNA, then we destroy that big messenger RNA, and now I don't make that protein anymore. Oh, that's kind of cool. Now, if it's small interfering, that's just the exact same concept, except you need like the entire RNA guide to match up with that messenger RNA, or at least that portion of the messenger RNA. And so basically it's just much more specific. So small interfering RNA is very, very specific. And there are lots of really nice therapeutic applications. So you can imagine if you had a patient who's making too much of a specific kind of protein, and maybe that's hurting them, then you could send in a small interfering RNA that's very specific, and it will disable that messenger RNA that's making that aberrant protein, and thus you can get rid of that protein in that person's cells. And so I want to end with this video. It's a really well done video from Nature, the journal, and it's going to talk about RNA interference, and it's going to talk a little bit about some of the therapeutic applications.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaut, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaut catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonauts and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.